Hello, everyone, and a warm welcome to the third week of our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia. I'll be your host for today's session. Myself and our wonderful team of co-organisers, Andreas, Julia and Wouter, in the groups of Judith Mank and Simone Imler at the Universities of British Columbia and of East Anglia, are very grateful for the massive support from the research community for this initiative. And we're delighted by the fantastic lineup of speakers we have planned for you. So thank you all for your engagement and the excellent set of talks that we have been honoured to host so far. Before I introduce today's speaker, I'd like to remind you all that there will be a Q&A session directly after the talk. So please post your questions in the dedicated Slack channel and upvote questions you'd like to hear. We aim to keep the whole session to 1 hour in length. So it is my great pleasure to introduce today's speaker, Professor Thomas Flatt, Professor of Evolutionary Biology at the University of Fribourg in Switzerland. Thomas's research explores the population genomic basis of adaptation and the evolutionary and genetic mechanisms underlying lifespan, reproduction, and aging in both natural and laboratory populations of the fruit fly Drosophila melanogaster. Most recently, Thomas's work focuses on the intriguing role of chromosomal inversions and supergenes in shaping adaptation to latitudinal gradients. Thomas also co-founded the European Drosophila Population Genomics Consortium, GWAS EU, and currently leads an exciting and active program of activities through this network. Today, Thomas will present his talk entitled The Genomic Basis of Adaptation Along Clines. So Thomas, thank you for accepting our invitation, and over to you.
Thank you very much, Elizabeth, for the very generous and kind introduction, and thanks a lot for having me. It's a great pleasure to be giving this online talk, and I will share my screen now with you and turn off my video so that I don't get distracted too much. So let's see. Okay, so what I would like to do today is to tell you a little bit about what we have recently learned about the genomic basis of adaptation along Clines, mainly using the fruit fly Drosophila melanogaster as a model system. So the term Cline, or the concept of Cline, was coined by Huxley in 1938 to describe gradual systematic changes of trait values or allele frequencies across space, for example, across latitude or altitude. And it thought that these gradients in trait values or allele frequencies are driven by underlying environmental gradients, for example, gradients in temperature or salinity. Many of these Clines are supposed to be driven by what we call spatially varying selection. And so this is illustrated here in the upper part of the figure, where we have some spatial variation in the selection coefficients or fitness values across the environment, resulting in step-like Clines or more continuous Clines. And so often, if we see such strong Clines, we think about them being driven by spatially varying selection. However, I should also point out that many of these Clines can be driven by or sometimes confounded by demographic processes such as population structure or admixture and so forth. So just observing a Cline doesn't necessarily always imply selection. And so selective or adaptive Clines need to be carefully distinguished from potentially confounding demographic factors. So here is an example of such a latitudinal Cline. Clines are very common from plants to humans. And here is an example from Arabidopsis thaliana, work done by John Stinchcombe and collaborators, published in PNAS in 2004, where John and his colleagues found a strong latitudinal Cline in terms of mean days until bolting, so a flowering time Cline. And they've been able to link this Cline in their work to a flowering time locus called Frigida. And in our own work, we have been interested mainly in studying latitudinal Clines in Drosophila melanogaster as a model system for learning more, understanding more about the genetics and the genomics of life history adaptation. So we've been working on this on and off for about 10 years. And most of our work has focused on latitudinal Clines in North America, but in some work we have also been looking at clines in Europe, in our recent work, and also in sub-Saharan Africa. So before I tell you about some of our findings, I shall like to acknowledge the people who have actually done the work. So first, in the upper left, we have Martin Kapoon, a talented former postdoc in the lab who is now a junior group leader in Zurich and in Vienna. And then two current postdocs, Ezra Durmas and Envel Kerdafrek, who have contributed phenotypic data that I'm going to show you in this talk. Importantly also, none of this work would have been possible without our long-term collaborator, Paul Schmidt from the University of Pennsylvania in Philadelphia. So Paul has been helping us a great deal, mainly also by giving us access to fly materials that he has been collecting across some of these gradients. Now, why are fruit flies an interesting model for understanding adaptation? And to explain this, I need to tell you a little bit about fruit fly demography. So Drosophila melanogaster is an ancestrally tropical insect that originated presumably down here in the region of Zambia and Zimbabwe, and then expanded around 10,000 years ago from southern Central Africa through the Sahara into the Middle East, and from the Middle East then colonized Europe, according to the most recent estimates by Sprengelmeyer et al., around 1,400 years ago. So this is a tropical insect ancestrally that essentially has spread around the world and then become cosmopolitan. So from the Middle East and Europe, this species has then migrated into Eurasia, then also into Australasia, colonized Australia. So for example, we have very prominent clinal gradients here along the Australian east coast, and it has also colonized Northern America roughly about 150 years ago, as we can tell. And the Australian gradient here along the Australian east coast is also around 150 years old, as far as we can tell. So this migration out of Africa led to these fly populations forming several latitudinal gradients that span often tropical, subtropical areas, as well as temperate seasonal areas. And so therefore, this system is a good one if one wants to understand relatively recent climate adaptations. So when this tropical insect migrated around the world and became cosmopolitan, it had to presumably adapt to new environments, mainly new climate zones. For example, seasonal climates, also sometimes characterized by harsh winters. Now, most of our work has been focusing on the North American latitudinal cline, which is depicted here on the left-hand side. So we have been studying in collaboration with Paul several populations along this cline, and the endpoints of this cline are kind of approximated by high latitude populations up in Maine and Vermont or in Canada, and on the other end, they're approximated by low latitude populations in southern Florida. So if we kind of look at the endpoints of these clines, we see, of course, that there's very different climates. So in Maine, we have strongly temperate, very seasonal climates and habitats, strong and harsh winters, which presumably can also lead to winter dieback of some of these fruit fly populations. Whereas in southern Florida, we have perhaps a more stable environment, a subtropical habitat. And we've been interested in studying this cline from a genetic and phenotypic perspective. As I've also mentioned on the previous slide, actually the first record of this species from North America dates back to 1875. So we think that the formation of this cline is a relative recent event. Now, why are such latitudinal clines interesting from the perspective of adaptation? And one of the main reasons is that we see at the phenotypic level many strong latitudinal clines for fitness-related traits, so-called fitness components. And so here on this picture, I'm showing you an example from the northern and the southern hemisphere. So here in this plot, you see the outline of the North American East Coast, and down here in the southern hemisphere, we see the Australian East Coast. And so, for example, if we look at one of the major fitness proxies, body size, we see that flies in low latitude populations are genetically smaller than flies up here in high latitude populations, where the flies are genetically larger. And if we look along the Australian cline, we see exactly the same cline, qualitatively speaking, with flies being genetically small in tropical Queensland, but being genetically large in places like New South Wales and Tasmania. And so this parallelism is already an intriguing feature of this system and gives us some replication, if you will, of these clinal patterns. Beyond body size, we also see strong clines for other fitness components, such as lifespan. So flies in Maine and Vermont genetically live longer than flies from southern Florida. And we also see that flies from high latitude populations are more stress resistant in terms of starvation stress. They're better at surviving cold stress. Interestingly, they're also better at surviving heat stress. And these flies in high latitude populations also have the phenotypically plastic ability to undergo what we call reproductive dormancy or diapause, which is a syndrome that is thought to be underlying their ability to overwinter. In contrast, flies from low latitude populations in southern Florida, for the most part, do not have this plastic ability. So when you stick them into these kind of like diapause conditions, which are low temperatures and relatively short photo periods, these flies from southern Florida don't go into dormancy. Flies from low latitude populations, on the other hand, are characterized by higher levels of fecundity and presumably also by more efficient larval growth and presumably also a higher larval competitive ability. And this already illustrates that flies along these clines have different life history strategies, with flies from high latitude populations apparently investing more into somatic maintenance and survival, whereas flies from low latitude populations might be thought of as investing perhaps more into reproduction and larval growth. Now, today, I would essentially like to address two questions. And the first one is, where in the genome do clinal variants map? And secondly, do these variants that we see in the genome, can we show that they contribute to some of these life history clines? This was the primary motivation of starting this work. Now, if we take a genomic look, as we have done here in this 2012 paper, where we have compared the genomes of flies from southern Florida, from a population in Pennsylvania here, 40 degrees latitude, and a population in Maine, we see that there's many strongly clinally varying SNPs in the genome. And here I'm just showing you 4 random examples of loci in the fly genome where we see such clinality. So for some of these clinal allele frequency gradients, so for example, for this gene here, we see very strong clinality of the alleles, going from 20% in Florida up to almost 100%. And so this is a quite common feature of many of these clinally varying loci. Some of them show very pronounced clinality. And given that we know that gene flow among fruit fly populations along these gradients is pretty strong, so these populations are strongly interconnected, and isolation by distance is actually very weak, it's tempting to think that many of these clinal gradients might be shaped by spatially varying selection. However, as you will also appreciate, in many of these loci, we see many clinal SNPs, and it's clear that many of these SNPs might just simply be dragged along. They might be hitchhiking and subject to genetic draft, because they might be linked, but they're not necessarily causative. Moreover, as I've mentioned before, some of these clinal gradients can also be potentially confounded by demography. What I should also point out is, in this paper in 2012, when we looked at the North American cline on a genomic level, we also compared our data to another paper from David Bigan's group, looking at the Australian cline. And if we look at the most strongly clinally varying genes in the fly genome, we see a significant overlap with the Australian cline. At the candidate gene level, that overlap is something like 30%, and it's highly significant. So that already tells us that, at least in part, the genetic basis of some of these clinal gradients might be shared across continents. Now, what happens if we take a more global look at the genome? And where in the genome do the most clinally varying SNPs map? To answer this question, we have more recently performed SNP-wise binomial GLMs using latitude as a predictor. And in the plot shown below, we have done this for 10 populations along the North American cline. What you see in this Manhattan plot on the x-axis are the different major chromosomal arms of the fruit fly, and on the y-axis we have a measure of p-value. For this analysis, we have kind of taken a very extreme outlier approach, and one could be much less stringent than we have been here. But what I'm showing you here are the top 0.1% of the most significantly clinal SNPs from these latitudinal regressions shown in red. So depending on how we do this analysis, we see that there's many strongly clinally varying SNPs dispersed or interspersed throughout the entire genome. So we see very pronounced peaks here, for example, and over here on 3L. But what is clear, and independent of the statistical details of how we do this and how stringent we are with the outliers, we always find a super strong enrichment here on the right arm of the third chromosome. And so in this particular analysis, actually 90% of the most strongly clinally varying SNPs map to the right arm of the third chromosome. This pattern had been noticed by us before in 2012 in terms of FST differentiation between Florida and Maine, and the year prior to that in this study of the Australian Cline, where they also found very strong FST divergence between, I believe it was Queensland and Tasmania. So what is special about this region on 3R? Well, it turns out that this region is spanned by a very well-known chromosomal inversion in the fly, which is called IN3R Payne. It's named after the person who first discovered this inversion, Fernandez Payne, in 1918. So chromosomal inversions are rare structural mutations where essentially we have a standard arrangement. This is kind of the standard terminology for a normal uninverted or non-inverted chromosome. And the inversion, this rare mutation, then results in a reversal of the gene order. The main property, the main genetic effect of such inversions is that they strongly suppress and reduce recombination when they're in heterozygote state. So in other words, if I have an individual with the standard arrangement chromosome, but the other homologous chromosome carries the inversion, then recombination is very effectively suppressed, especially at the 2 breakpoints of the inversion. So these inversions have been first discovered in 1919 by Sturtevant, also in the fruit fly, and they have been studied for a long time cytologically, and they have this pretty amazing property that here in these salivary gland preparations, you can kind of see these loops, and these loops exist because the loop essentially tries to maximize the pairing of the sister chromatids in meiosis. Because there is this reversal in gene order, there can be massive problems during cell division and meiosis, and so this loop kind of tries to maximize the homologous pairing of. I hope you can still hear me.
We lost your video connection.
Okay, so now it's fine again?
No, I don't see anything.
Oops. Do you see it now?
I see you, but not your screen.
Okay. Now you should see the screen, right?
Yeah. Perfect.
Okay, so what we see here are these heterozygote loops that can be diagnosed using cytological karyotyping, but nowadays we actually have PCR markers and diagnostic SNP markers to diagnose the presence of these inversions. So what do we know about this inversion polymorphism? We know that it's very large. It's about 8 megabases. That's about 30% of the right arm of the third chromosome. This inversion is of ancestrally African origin. It's not super ancient. It's about 130,000 years ago. We know that it's polymorphic, so not every fly carries it, and it has different frequencies in different populations, so it segregates. It's cosmopolitan, as we have known for some time, And already work in the 70s and 80s showed that it forms strong latitudinal clines on multiple continents. And we also now know that it's monophyletic, and this is illustrated in this graph here, which is a neighbor joining haplotype network that Martin Capun constructed using data from 480 fly lines. So the edges here in black denote fly lines that carry the standard uninverted arrangement, and the lines here in cyan blue denote lines that carry the inverted chromosomal arrangement. And you can clearly see that the inversion is of monophyletic origin. You also see some geographic substructure. So here at the tips of these lines, you see the different color codes for Africa, Europe, USA, and Australia. And so you see that within a karyotype, there's some geographic substructure. And that's true also for the non-inverted standard arrangements. But the big signal here really is karyotype, and it's clear that this inversion is of monophyletic origin. Oops. Now, it's tempting to speculate that since the majority of the most strongly clinally varying SNPs map to this inversion, and because many fitness components exhibit very strong clinality, that this inversion might be one of the major drivers, if not the major driver, of clinal life history adaptation in this system. So what would we expect if we are dealing with an adaptive inversion? So for example, we would expect that the inversion cline should deviate from neutral expectation. We might also predict that the inversion polymorphism should be stably maintained over time, presumably due to some balancing selection. And there should also be some genomic signatures that are consistent with selection on the inversion. And perhaps finally, we might expect that the inversion affects clinally varying fitness components and therefore contributes to the phenotypic clines. And so what I would like to do now is to walk you through some evidence that this inversion system that we have been studying fulfills some of these criteria. So first off, IN3R Payne is strongly clinal on multiple continents, and this has already been investigated in the 70s and in the 80s. And so this figure here shows you a recent meta-analysis that Martin and I performed in this 2019 paper, where we've taken 34 data sets spanning about 50 years of observations. And so what this data shows is that this inversion is strongly clinal along the North American East Coast, always being absent in Maine and Vermont, but being rather common in southern Florida, reaching a frequency of 50%, or some places even higher, sometimes 70 or 80%. We have also recent unpublished data showing that the inversion has a more shallow cline in Europe, but is also significantly clinal. We see the inversion cline along the Indian subcontinent, which is also spanned by both tropical and temperate regions. We see a strong latitudinal cline in Japan. And then for the southern hemisphere, there's good data, as I've mentioned, from the Australian East Coast, where we see the same qualitative cline upside down. Now, we can also more formally try to see whether we can come up with evidence whether selection actually maintains this inversion cline. And to do so, we've investigated the frequency of this inversion from pool-seq data using highly specific inversion specific SNP markers that are diagnostic for the presence or the absence of the inversion. So here in the upper plot, you see again decline going from 40 or 50% down to 0 as we increase latitude. We see a little bit of seasonal fluctuation, but not really much as compared to other inversions. And using this data, we can also look at temporal stability by comparing our genomic data in red to cytological karyotyping data. And this data essentially shows that the clinal slope has been maintained over a time span of approximately 40 years. Now, we can also look at the clinal slopes and compare them to neutral expectation by looking at 10,000 SNPs in short introns, which are presumably evolving neutral. And so these intronic SNPs, the cline they're forming is shown down here by this dashed line. And so when we look at these presumably neutral SNPs, we see a very shallow cline. So there's very weak isolation by distance, presumably because gene flow is very strong. And the inversion cline significantly deviates from this pattern that we would expect under neutrality. We have also looked in this study at admixture because it has been reported then that low latitude populations might be subject to admixture by African genotypes, and high latitude populations in Maine are subject to admixture from European genotypes. And such differential that might confound the inversion cline or contribute to it. But our back of the envelope calculation shown here as this dashed line shows that the inversion cline significantly deviates from admixture. So we have pretty strong evidence that this inversion polymorphism is actually maintained by some sort of spatially varying selection. Now, what would we expect in terms of the signature of genomic divergence? So these plots here show you the divergence between non-inverted and inverted chromosomes, divergence measured in FST or DXY along the chromosome, and here to the left and the right, these dashed lines denote the breakpoints. And so coalescent theory by Rafael Guerrero and Mark Kirkpatrick has derived coalescent expectations for inversions that are older than NE generations. So what pattern of divergence would we expect under neutrality? We would expect to see very strong divergence at the two breakpoints, because at the breakpoints, recombination is absolutely or completely suppressed. However, if the inversion is sufficiently old, there can be some gene flux, some genetic exchange in the center of the inversion away from the breakpoints, genetic exchange between the inverted and non-inverted chromosomes due to gene conversion and double crossover events. So essentially would get kind of a suspension bridge pattern under neutrality. I should also point out though, that the pattern will look qualitatively similar if selection, positive selection, would act on mutations in the two breakpoints. However, if we look at the scenario of local adaptation, where an inversion comes along by chance and captures a pre-existing locally adapted haplotype, we might expect to see some strong peaks of divergence in the center of the inversion away from the breakpoints. And the idea here is that selection maintains these presumably adaptive peaks in the center of the inversion despite homogenizing gene flux. So how does this pattern look like for our inversion? So we have looked at this using pool sequencing data, where we have done pool sequencing of the two homokaryons that we have isolated in a relatively large number. And we've done this in our study for North American flies and European flies. The comparisons in green and red show the comparison between the inverted chromosomes and the standard chromosomes, either from the same population, here Florida inverted versus Florida standard. For Europe, we have compared inverted chromosomes from Portugal to standard chromosomes from Portugal from the same population. And here in red, we see the contrast between the inverted and non-inverted chromosomes across geography. So between the endpoint populations along the Cline, here Florida versus Maine, and here Portugal inverted versus Finland standard. And the blue comparison shows you the genetic differentiation between the standard chromosomes from the different places. So what you see here for the red and green comparisons are, first of all, you see very nicely the two breakpoints, and you also see very notable pronounced peaks in the center of the inversion away from the breakpoints. And notably, we see qualitatively the same pattern, a very congruent pattern in Europe. So this is pretty remarkable. And so at least qualitatively speaking, these peaks in the center could be consistent with this local adaptation model. Now, using phase sequencing data of haplotypes using a so-called hemiclone approach, we can also look at linkage disequilibrium. So for this, we can't use the pool sequencing data. We need to know the sequence in the haplotypes. So if we look at LD between inverted chromosomes, we see that LD is pretty much absent. There's no strong LD, as you would expect, because inverted chromosomes have free genetics exchange between them. And we see the same thing for standard arrangement chromosomes. So all the LD we find here in this triangle is from the comparisons between inverted and standard chromosomes. And this, of course, nicely fits the fact that inversions suppress or reduce recombination in heterozygous state. Now, we can also look at LD as a function of the genomic position and as a function of these peaks. So here again, we have the breakpoints, and here we have these pretty big peaks in the center of the inversion. So there's like 2 big peaks here, there's some smaller peaks here away from the breakpoints. These central peaks are around 200 to 300 kb long, so they're very large. And using these LD analyses, we can show that these center peaks are in strong LD with each other, and also with the breakpoints, and also with some smaller peaks. And so this is also sort of qualitatively consistent with this Kirkpatrick-Barton model of local adaptation, where selection maintains strong divergence in the center of inversion despite homogenizing gene flux. However, from this pattern alone, of course, we can't prove that these central peaks are due to selection. They could also be due to the stochastic nature of the coalescent process, due to population bottlenecks, and so forth. But nonetheless, we were interested in taking a closer look at candidate genes and SNPs in these phase data for the center peaks of the inversion. And we have also used the GWAS approach to identify potential candidate genes in these peaks. So in the GWAS approach, which we have applied to pool-seq data, we essentially used linear regression using inversion frequency as a predictor and did this analysis genome-wide. So if you do this GWAS using the Payne frequency as a predictor, you see lots of signal here throughout the genome. But of course, the strongest signal is here on the right arm of the 3rd chromosome. And this is kind of like an unbiased genome-wide analysis. And what you see here are these marked breakpoints that are traced out by this GWAS analysis. And you also, with some imagination, can see here these center peaks. And so these phase data and this GWAS analysis has given us quite many interesting candidate loci that might potentially explain the adaptive nature of this inversion. So we find many interesting major candidate loci that underlie effects on body size. And we know this from mutant and transgenic analysis that these are candidate loci for these traits. So for body size, for example, the insulin-like receptor, there's also candidate genes for reproductive dormancy and for lifespan that are all found in these center peaks. I should also point out that about 90% of the inversion-specific top candidates in these peaks also are polymorphic and strongly clinal in the Australian data set. So there seems to be some shared basis of the clinality that we see here. More recently, we have performed RNA-seq to see whether and how this inversion might affect gene expression and to learn more about potential candidate loci. So what I'm showing you here are some preliminary data that we have of differentially expressed genes within the inversion, shown here in red. But we also see effects on differential gene expression on the other chromosomal arms. So the presence or absence of this inversion really has genome-wide effects on gene expression that are a function of this karyotype. This has actually also been seen for other inversions in this recent paper by Said and Russ Corbett-Dettig, and in a study by Lavington and Kern for other inversions where they also saw genome-wide effects. We also find a significant overlap between the genomic SNP candidates and the candidates that are differentially expressed in the inversion region. So using such approaches, we hope to learn more about the underlying loci and SNPs that might potentially explain why this inversion might be adaptive. But that will, of course, require much more fine-scaled work. Now, another major question is whether this inversion actually affects any fitness components that I've already told you are often very strongly clinal. And to answer this question, we have performed a series of assays where we have isolated isochromosomal lines that carry 2 copies of the standard arrangement from Maine, and where we have isolated isochromosomal homozygous lines carrying 2 copies of the inversion or 2 copies of the standard arrangement, and then phenotype these lines in the lab. So we have looked at a couple of fitness components, including developmental time, viability, that is egg to adult survival, several proxies of body size, lifespan, and a couple of stress resistance traits. So what do we find? The first result is that we see that the inversion confers small body size. And this example here is for wing area, but also holds for other proxies of body size. The bars in red denote the inversion homokaryons from Florida, the bars in light blue standard arrangement homokaryons from Florida, and then we have the standard arrangement flies from Maine, and we have looked at this in both females and males. And what we see here is that the inversion homokaryons in both females and males are smaller than the standard arrangement homokaryons. You also see, despite the difference in geography, so these are flies from Florida and these are flies from Maine, that there's no difference here in terms of geography. The data really cluster nicely together by karyotype. So it's also interesting to note that using a different approach, an association study by Ari Hoffman and his collaborators in 2006, essentially the same effects were found in terms of wing size for the Australian cline. So we believe that at least for two of these clinal gradients in Australia and North America, that we see parallel effects on body size. I should also remind you, I've already told you at the beginning, that flies from high latitude populations tend to be larger than flies from low latitude populations. So what we see here for the karyotypic difference nicely matches the overall clinal pattern in terms of phenotype. We can actually even use these data to ask what is the relative contribution of this inversion polymorphism to the total phenotypic wing size cline. And if we do a back of the envelope calculation, our calculation suggests that about 50% of the total cline, in this case of wing area, is explained by this inversion polymorphism. So a rather substantial effect. We have also looked at several wing traits. For example, we have looked at something that is called wing loading. Wing loading is essentially the ratio between wing area and total body size. And we see that wing loading is lower for standard arrangement flies. And this is actually also the directionality that we would predict from the overall phenotypic cline. So flies in Maine are known to have reduced wing loading as compared to flies in Florida. So that means they have relatively larger wings relative to their body. And this has been shown in some papers to have potential effects on flight ability and lift under cold weather conditions. Between the different karyotypes, we also see some subtle differences shown here in terms of wing shape deformation with expansions and contractions. But the significance, the functional significance of these wing shape deformations is perhaps a little bit less clear. Now, what about other fitness components? Another component of fitness that we have looked at is lifespan of adult flies under different temperatures. And here we see that the inversion flies, the inversion homokaryons, significantly live shorter than the standard arrangement homokaryons, both at 18 degrees and at 25. And this is true for both females and males over here. So the pattern, the karyotypic differentiation is perhaps a little bit more clear at 25, where the two standard arrangements shown in blue and red cluster very nicely together. And at 18, you see that the survivorship curves are a little bit more spaced apart. So this could potentially indicate that there is some difference in geography superimposed on these karyotypic effects. So in red, we have the standard arrangements from Florida, and the blue survivorship curve shows you the standard arrangement from Maine. So with some fantasy, we can see here that maybe the differences between the karyotypes are superimposed by some geographic difference for lifespan. We have also looked at some stress resistance traits. And so here I'm showing you the example for cold susceptibility. When we expose flies to 90 minutes at 4 degrees Celsius, and we look at survival upon this cold shock, we see that flies that developed at 25 degrees Celsius survive much less well if they carry 2 copies of the inversion as compared to the standard arrangement flies from Florida and Maine. So the inversion seems to confer cold susceptibility. Now, you might have noticed that all the fitness component values I've shown you so far tend to go into kind of a negative direction for the inversion. So the presence of 2 copies of the inverted arrangement seems to reduce the values of these fitness components. These flies are relatively small. They don't seem to be super cold shock resistant. They live shorter and so forth. But you should remember that the experiments I've shown you here are based on isochromosomal lines where the flies are essentially homozygous for the chromosomes. So we essentially isolate the single wild-type chromosome carrying either the inversion or being standard arrangement and then making 2 copies of that chromosome. And so that means that these flies might be potentially subject to inbreeding depression, and we might be exposing lots of recessive deleterious alleles. So more recently, we therefore wanted to look at heterozygous material, where we cross fly lines carrying either karyotype from different families to generate heterozygous material. And using this material, we have looked at both homo as well as heterokaryons. On the previous slides, I've just shown you the homokaryons, inverted versus standard. But what about the heterokaryons? Most of the action might actually be in these heterokaryons, where recombination is absolutely suppressed. So here we don't have so many data yet, so this is work in progress, and we're currently working on large diallele crosses to generate such material. And here I'm showing you a preliminary result for egg to adult survival or viability, where we see that the heterokaryons seem to have a higher viability, statistically significantly higher viability than either the inversion homokaryons or the standard homokaryons. And so this could be quite promising because it might mean that at least for this trait, there might be some sort of heterosis or heterozygote advantage. Now, it's pretty unlikely that in such a large chromosomal inversion, this would be driven by a single overdominant locus. We can't exclude this possibility. An overdominant mutation might arise in the breakpoint, but it's perhaps more likely to think that such a heterozygote effect might be the effect across many loci, so that what we might be looking at here is some kind of multi-locus heterosis. This is also sometimes called associative overdominance or pseudo-overdominance, and this is a possibility that we're very interested in exploring now, because it might potentially explain why this inversion polymorphism is maintained locally at relatively stable frequencies. So in summary, I've shown you that most clinely varying SNPs in Drosophila and melanogaster map to a very large chromosomal inversion, which itself is known to be clinal, actually has been known to be clinal for a long time, and seems to be subject to spatially varying and also balancing selection. It's interesting that this system shows some parallel clinality, at least qualitatively, on multiple continents. So on pretty much every continent that has been examined carefully, we see the same qualitative clines, often also quantitatively speaking in terms of the clinal gradients. And the inversion always tends to be frequent in tropical, subtropical areas, but pretty much absent as far as we can tell in temperate seasonal areas. And so it will be interesting to explain why is it good to have this inversion polymorphism at some intermediate frequency where it's warm, but why should it not be good to have it in areas where it's cold, where you have pronounced winters and seasonal climates? I've also shown you some data to suggest that this inversion has some pleiotropic effects on body size, lifespan, and a bunch of stress resistance traits. And so we could argue that based on the fact that these traits are very complex quantitative traits, that these effects are unlikely to be caused by a single locus, but of course we can't completely rule that out yet. And it's notable that these effects of the inversion polymorphism on these fitness components, that these effects go in the same direction as the overall phenotypic clines known for these traits. So we think that this inversion might somehow represent perhaps a supergene, a set of tightly linked loci that affect multiple complex phenotypes relevant for adaptation and fitness, and that the inversion kind of like binds together these loci inside the inversion. However, how exactly this inversion is being maintained remains to be elucidated in much more detail. So I hope to convince you that we have an interesting case of clinal adaptation and its genetic basis. We believe that clinal adaptation in this species is predominantly driven by this inversion, but obviously many fascinating and also very difficult questions remain completely open. And this has been very nicely summarized in a piece by Mark Kirkpatrick and Andy Kern in 2012. With the very cool title, Where's the Money? Inversion Genes and the Hunt for Genomic Targets of Selection. And what Kirkpatrick and Kern conclude here is that despite many decades of research on inversions, going back to Sturtevant and Morgan and Dobzhansky, we still know very little about which of the genes these inversions carry are the actual targets of selection, or how the polymorphisms themselves are maintained. So using genomic tools and population genetics and also phenotypic assays, we hope to contribute to a better understanding of this issue. So what's next for us? Currently, we're planning population cage assays in outdoor mesocosms in collaboration with Paul Schmidt's lab. So this is a picture of Paul Schmidt's field facility, where we would like to do some large population cage assays to look at frequency-dependent selection and over-dominant selection under field conditions, and to do similar assays in parallel in the laboratory. As I've mentioned, we're working on extensive diallele crosses, especially quantifying fitness components for the heterokaryons to elucidate the potentially over-dominant nature of this inversion. And we also have a major interest in trying to use CRISPR-Cas9 to look at some of the most promising clinally varying SNPs in the center of the inversion away from the breakpoints. And so this is all ongoing work that's going to take a while. These problems are not necessarily trivial, but we hope that we will learn more using these approaches. So finally, I shall like to thank the people from my team. Some of them are shown here in this recent picture, also our funding support, and thank you very much for listening.
Fantastic. Thank you very much, Thomas. That was a really interesting talk. Lots of very interesting data presenters and a curious story for sure with these latitudinal gradients and the role of this inversion polymorphism. So I thought I'd start with a question first of all, and we also have quite a few now coming in our Slack channel as well. So you mentioned when you were looking at the inversion's effect on fitness traits, that about 50% of the phenotypic Cline was explained by the inversion polymorphism, which is obviously a large percentage. But I was quite curious if you had some ideas about what could explain the remaining percentage and whether it could be many regions of the genome or perhaps particular loci or other even inversions which could have some effect on the fitness traits as well.
Yes, absolutely. So it's clear that there's multiple loci and SNPs that are clinally varying that contribute to these size clines. So just last year, we've published a paper where we looked at the clinally varying polymorphism that is not spanned by this inversion in an insulin signaling gene, which also makes a pretty strong contribution to this Cline. There's also work by Paul Schmidt on indel polymorphism in the insulin-like receptor. This is a gene that is spanned by the inversion, but this particular indel polymorphism doesn't seem to be linked to the inversion itself. And this gene or this polymorphism also contributes to the Cline. So I would say body size, as one might imagine, is a highly polygenic trait. So I don't think it's only going to be the inversion. It's going to be many other things. And one also has to take these estimates that I've presented with a little bit of a grain of salt, because in order to make this estimate and to estimate the relative contribution to the overall Cline, we are in a way isolating the inversion and of course taking it out of its natural genomic context. There could be other interactions with other inversions, with other parts of the genome that we don't know about. And some of these interactions might actually undo some of the effects that we are estimating. So essentially, if you have kind of like some suppressing epistasis and so forth, you might get some effects. So for the clinal polymorphism in this FOXO gene we've looked at last year in an evolution paper, we've estimated that it might explain 13% of the Cline, which also seems a rather large effect size. But by doing this experiment, we actually have to isolate the polymorphism and in a way take it out of its natural genomic context. And so it's not clear whether these absolute estimates, whether they would not be undone to some extent by interactions with other loci.
Right. Yeah, that makes sense. That's really interesting. So we'll move on to some questions now from our audience, which have been posted in Slack. So our first question. So the question asks, is the inversion mutation rate temperature dependent, meaning that the mutation rate is higher in warmer climates versus colder?
That's an interesting question. I don't know about this. It could be, but I doubt it. But I'm not really sure. I don't think so.
Okay.
I mean, what I could add maybe is, I mean, so there's like several mechanisms that can give rise to these inversion mutations. And so it's clear that in the genome, there are some weak spots, especially also spots with a lot of repetitive DNA sequence where some spots of the genome are more prone to breakage and might give rise to inversions. But whether that's strongly temperature dependent, I don't know, to be honest.
Okay, it's interesting. So we've got a few questions which are related to reproduction and whether there's an effect of the inversion on reproduction, or if there's any data on that yet, and any clinal variation, I guess, in that trait as well.
Yes. So, I mean, what we know from overall phenotypic clines, at least from North America, and that's mainly Paul's data, is that there is clinality in terms of fecundity and fertility, and things like ovarial number and so forth. Also maybe linked this syndrome of reproductive dormancy that flies in northern populations can express, but not in low latitude populations. But for the inversion itself, we haven't actually investigated fecundity yet, or fertility. And so I think that's going to be a very interesting thing to do with these diallele crosses once we have a larger number of heterokaryons that we can look at. But it seems clear that the flies from low latitude populations in southern Florida, they tend to be more fecund. Another thing that's different is that these flies in southern Florida, they might be a little bit more efficient in terms of their growth, but that also needs more investigation. And so one model that we have is that these flies might also be better larval competitors, but we need to do more experiments to really firm this up.
And could that be linked in with the standard metabolic rates at all, do you think? And whether there could be differences in the standard metabolic rate between populations with and without the inversion?
Yes, I mean, so sometimes there's also correlations between metabolic rate and body size along some of these Burton Clines. I don't think this has been properly investigated in flies. I mean, we haven't done too much physiology. We have looked a little bit at metabolic storage, like fat content and things like this. It seems generally that flies in manes seem to invest more into somatic maintenance, and maybe flies in southern Florida are more prone to have higher energy expenditure more quickly. But I don't know in terms of metabolic rate. But something that, of course, could be looked at in the lab.
Yeah, that's great. Does the inversion frequency vary between males and females, do you find?
Not as far as we can tell. Not as far as we can tell. Yeah. So okay. I don't think there's a difference between the sexes in the frequency. I mean, many of our pool-seq data are from males actually, but other data are from females. So I don't think there's a pronounced sex difference. So I don't think there's strong interaction with sex-linked genes in terms of this inversion. And also in terms of, I mean, I've briefly alluded to this. I mean, this is not directly linked to the issue of sex linkage, but many inversions also show some seasonal fluctuations. But for the one that we're studying here, we don't really see a lot of seasonality.
Ah, okay. Do you see much dispersal of the Drosophila populations across a cline? So could they move between different climatic conditions, and could that influence the traits to it?
Yes, so we think that there must be very strong gene flow along some of these clinal gradients, especially in North America. And this could, of course, also be further influenced by human movements and food transports and things like this. And so if you look at presumably neutral markers in short introns or at fourfold degenerate sites, you tend to see very shallow Clines. So there seems to be generally very shallow isolation by distance, indicating perhaps that gene flow is very homogenizing across these gradients. And so against this backdrop or background, it's very interesting if you see very steep Clines. It's quite unlikely that these would be explained away by isolation by distance and so forth. But these flies definitely move around. People might move them around. And very interesting is this phenomenon that was investigated by a couple of groups, including by Alan Birkeland, showing that there can be some differential admixture at the Cline ends. So in southern Florida, you have influx of African genotypes, presumably through the Caribbean, into southern Florida. And in Maine, for whatever reason, there's a predominant influx of European genotypes. And so as I've mentioned in my talk, this differential admixture could in principle also generate Clines. And so we don't think this really confounds this inversion Cline, but this can have genome-wide effects for sure.
Ah, great. That's interesting. So I think we'll just have one or maybe two more questions before we finish. So there's a question which says, I know that at least Drosophila subobscura also have inversions, which are also associated with Clines. Do you know of more species Clines where the solutions also involve, to some extent, inversions?
Yes, so there's lots of Drosophilates that have inversions, also lots of other insects. There are some Drosophila, though, that are pretty much inversion-free, for example, Drosophila simulans and Mauritiana and Seychellia. So there's some element of historical contingency or idiosyncrasy, perhaps. But in Drosophila pseudo-obscura and subobscura, there's very strong Clines, and these species harbor many inversions. In pseudo-obscura, there's very good evidence that some of these Clines have remained stable for more than 40 or 50 years. Dobzhansky studied them in pseudo-obscura extensively. So he reported seasonal fluctuations that were repeatable over years, perhaps indicating some temporally varying selection being at play, also strong altitudinal gradients for some of these inversions. And also in subobscura, this is the case, that there's very strong inversion Clines that presumably contribute to climate adaptation. And some of these Clines are thought to have shifted in their clinal slopes and intercepts due to global warming. So these inversion Clines are a very common phenomenon.
That's interesting. And I guess it will be interesting to see with the effects of climate change and changing climates across these Clines, how that affects the fitness traits and the effects of the inversion going forward. But that's great. I think that's all we have time for today. But thank you very much, Thomas, for a really fascinating talk and some excellent answers to the questions in the Q&A session. Thank you.
Thank you very much again, Elizabeth, and thank you everyone for listening.
Thank you. So before we finish, I'll just remind everyone about our next talk, which will be coming up this week on Wednesday, from Professor Sarah Otto, who will present an intriguing talk on narratives, theories, and models in evolutionary biology. So we hope you can join us on Wednesday, same time and same place. And in the meantime, check out our updates on Slack and our Twitter feeds. Thank you for taking part, and until next time, stay safe and see you again soon. Goodbye.